The overall goal of this protocol is to describe the fabrication of a flow-through tryptic microreactor that performs fast enzymatic digestion of proteins for enabling protein identification with an electrospray ionization mass spectrometer.
The method can help elucidate the amino acid sequence of isolated and purified proteins to support downstream characterization of structure and function.
The main advantage of this protocol is that it is fast, cost-effective, and prone for integration into workflows that make use of microfabricated platforms.
To begin, use a glass capillary cleaver to cut the larger glass capillary tube to a length of 7 to 8 centimeters and the smaller one to a length of 3 to 5 centimeters. Use a microscope to verify that both capillary ends have a clean, straight cut without any protruding burrs. Insert the smaller capillary about 6 millimeters into one end of the larger one. Then, use a Q-tip to apply a small droplet of glue around the junction of the capillaries and let the glue cure overnight at room temperature. Next, cut the inserted capillary to a length of about 10 to 15 millimeters. This end will act as the electrospray ionization emitter. Insert the opposite end of the larger diameter capillary into a 1/32 inch PEEK tube that has been pre-cut to between 4 and 5 centimeters in length and connected to a PEEK union. Then, insert and tighten a 5 centimeter long piece of 1/16 inch PTFE tubing into the opposite end of the union.